Okay. So I hope everybody can see this. So thank you, Mara, for the introduction. And I want to thank everybody at the Genome Writers Guild for organizing this conference and giving me a chance to speak to you all today. I'm very excited to kick things off. So get into presenter mode. So today my talk is called Prime Editing, Precise and Versatile Genome Editing Without Double-Strand Breaks or Donor DNA. And as Mara mentioned, I'm a postdoc in David Liu's lab at the Broad Institute, where I've done all of this work over the last couple of years. So I want to start off by just introducing to you all sort of the distribution of the types of mutations that cause disease in humans. And so on the left is a pie chart depicting the different types of genetic changes, for instance, point mutations, deletions, and duplications, and sort of the makeup in different diseases of how often these occur. And these lead to common diseases that we're all familiar with, like hemochromatosis or sickle cell disease or cystic fibrosis. And I think with the emergence of genome editing as a potential therapeutic, it's very exciting that we now can see in the not too distant future the possibility of correcting directly in a patient these different classes of mutations that give rise to the pathology. And so for me personally, this was a very motivating feature for the work that I'll be telling you about today. So obviously when Cas9 nuclease came onto the scene, sort of the field of genome editing really exploded. The thing that makes Cas9 so special, as you probably are all aware, is that it is programmable by a guide RNA molecule. And the sequence of this guide can tell Cas9 nuclease where to bind to DNA and where to cut it. And Cas9 cuts with two individual nuclease domains, a Rev-C domain and HNH domain. And it also requires this protospacer adjacent motif or PAM sequence to exist close by to where this guide RNA will bind. And once Cas9 recognizes its target DNA sequence, it can cut both strands of the DNA double helix and generate a double strand break. And we've known for quite some time that double strand breaks can be exploited through numerous cellular repair pathways to make genetic modifications. So the two major arms of repair that we think about are end joining and homology directed repair. So in the end joining pathway, in the absence of any type of donor DNA, these double strand breaks can create these indel products, which are mixtures of insertions and deletions, which are predictable at times, though not necessarily controllable. In some cases, you observe high yields of particular repair products, though more often than not, it's a quite diverse product distribution. Alternatively, you can use these breaks and insert DNA at the site of the break if you provide some double-stranded DNA that can be ligated in at that break site. So the sort of knock-in of end joining, using end joining, can be used to put in relatively large pieces of double-stranded DNA. However, the orientation of this insertion isn't always controllable. A more precise mechanism of repair is homology-directed repair or homologous recombination. And by using HDR or homology-directed repair, a donor DNA template can specify the edit that you would like to install. So for instance, single-stranded donor DNA can be used to install very precise local changes like base pair changes or small insertions and deletions. Alternatively, you can use larger DNA cargo, such as double-stranded DNA, pieces that contain entire genes or even larger pieces of DNA. And this can be inserted very specifically at the break site. So in theory, HDR provides everything you really can think of in terms of how you would might want to fix those pathogenic mutations I showed on the previous slide. However, the unfortunate reality is that nucleases, while they can direct these types of repairs in certain cell types, many cell types are not, don't have active homology-directed repair pathways. And therefore, more often, a double-strand break will go down the end joining pathway that leads to these indel byproducts. So only in some cases would that be an appropriate outcome for treating a disease. And I would say the majority of the time, what you require is a more precise change that might just not be possible with nucleases. So where have things gone since nucleases? So actually, in the last few years only, the base editors sort of emerged A lot of this work was pioneered by folks in David Liu's laboratory, and two major base editors have been established to date. The C base editor, which converts cytidine bases in DNA to thymine, as well as the A base editor, which carries out an A to G transition mutation. So these two classes of mutations actually account for about 30% of that pie chart, so a very large number of those changes. And more recently, just in the last week, actually, there's been reports of C to G base editors, which is a very exciting development. And this has now allowed base editors to enter into this transversion class of point mutations. But nonetheless, there's still the remainder of this pie, which cannot be effectively addressed by base editors, and certain cell types, homology-directed repair will just not be functional enough to carry out these changes. So this was the main motivation for the work that I'll be telling you about, is how can we theoretically install so that we can study these disease processes and correct these types of mutations so that we could potentially develop new therapeutics for human genetic disease. So the basis of the idea of how we might go about this really comes back to what makes the CRISPR-Cas9 system so special. And from my standpoint, it's the fact that this is a programmable DNA binding nuclease. So the spacer region of the guide RNA is a 20 nucleotide segment that, if it matches the target sequence, allows Cas9 to efficiently bind and cleave the DNA. So this programmability that's contained within this RNA molecule is really the special feature. So sort of prompted me to ask the question, can this guide RNA also carry an editing template? So in the same way that the spacer programs the target site, can this guide RNA, some other portion of it, specify the edit that you would like to make at that DNA target site? So if you're going to use RNA, of course, to program an edit in DNA, you need a mechanism to transfer that information from RNA to DNA, and that's reverse transcription. So in thinking about how this might work, luckily, nature has provided us with an example of how to carry out such a mechanism or a reaction. So I think one of the most prominent examples is the line class of retrotransposons. And for those of you not aware, the line elements make up roughly 17% of the human genome. So they're very successful retrotransposases. And the way that they work is they actually encode a protein that is a fusion between a AP endonuclease domain and a reverse transcriptase enzyme. They bind to relatively lenient target sites in the genome and create a nick in one of those strands of DNA. And then they use their own mRNA as a template to carry out reverse transcription using the genomic DNA as a primer. And in doing so, they're able to reverse transcribe directly into the genome their entire coding sequence, and through subsequent events, which are not actually fully understood, that DNA can be encoded at that site of cutting, and the retrotransposon has successfully made another copy of itself in the genome. So what if you're not trying to make this type of change, though? We're not very interested in making new transposases, but if you use the same principle and the concept that you can use this target primed reverse transcription to directly write new DNA at a specified target site, perhaps you could write just enough that covers an edit that you'd like to correct or install in the genome. So the question is, how can we use the very powerful programmability of Cas9 nuclease to bind any site in the genome virtually that you'd like and carry out this mechanism of target primed reverse transcription? If we were able to do that, you could imagine that we'd be able to generate this intermediate of DNA that has this new extra strand, which we call a 3' flap, that contains edited sequence. And in addition to the edited sequence shown in red, it also has some homologous sequence to the genome shown in black that would assist with DNA repair. So how might we do that using Cas9? So first, we wanted to take the nuclease and turn it into a nickase, because we don't want to make double strand breaks. And as shown on the mechanism on the left, we only actually need to nick a single strand to start writing new DNA into the genome. Secondly, we thought to fuse reverse transcriptase to Cas9. This is the enzyme that will carry out DNA polymerization using the guide RNA as a template. And so if we want to use the guide RNA as a template, we thought we could simply extend on either end, the 5' end or the 3' end, a template for reverse transcription that would allow you to program the edit that you'd like to make. So this is what we envisioned. And if you were able to make this intermediate of 3' flap sequence that contains the edit, we hypothesized that that strand could invade the adjacent DNA and displace a 5' flap sequence, which was the sequence, the DNA that was originally present in the genome. And this intermediate very much resembles intermediates generated during long patch base excision repair, as well as lagging strand DNA synthesis. So hundreds of millions of Okazaki fragments are generated every cell division. And in doing so, the DNA replication machinery generates 5' flap structures that are very efficiently cleaved. And then the nicked DNA is ligated. And this process is carried out by FEN1 endonuclease and other assisting enzymes. So we thought that this might be a mechanism by which we could install and replace that strand. So to generate a heteroduplex of DNA where one of the strands is edited and the other is not, then either DNA repair or replication of that strand could install the edit permanently into both sides of the double helix. So that was the mechanism and principles that we were going after to develop a new genome editing strategy. I'm going to skip all of the in vitro biochemistry experiments and other things we did to see if the system could work on a molecular level, sort of jump right into getting it working in mammalian cells. So what we ended up using as our editor, which we call prime editor or PE1, is the fusion between the marine leukemia virus reverse transcriptase enzyme and a Cas9 nickase. It's an H840A nickase, so that it nicks the strand that is not bound to the guide RNA, so the non-target strand. This is also the PAM-containing strand. And then we designed these prime editing guide RNAs or PEG RNAs that contain the same spacer sequence as a regular guide, but in addition to that have a 3' extension that carries the editing template. And so for the very first step, it's similar to other Cas9 mechanisms. The Cas9 PEG RNA complex will bind to target DNA, and it's going to nick the non-target strand. Fortunately, this non-target strand is quite flexible and generates an R loop of single-stranded DNA. So the hypothesis was, and this was supported by work that was previously done by the Corn Lab, that this single-stranded DNA would be flexible and available for action by reverse transcriptase enzyme. So the second step is that we have the primer DNA annealed to this extension of the guide RNA. So that region we call a primer binding site. And now this complex is perfectly poised to undergo reverse transcription of the edit encoding template that's just downstream of this primer binding site. So that reverse transcription process will polymerize directly into the genomic DNA, new DNA sequence that encodes the edit. So that was what we were going for. Initial attempts actually in mammalian cells were not successful using this system. However, what we quickly learned was that sort of the conditions required for our in vitro biochemistry experiments were not quite the same as what was required in mammalian cells. And what we simply had to do was extend this primer binding site sequence to be a bit longer so that the DNA primer could more efficiently anneal and initiate reverse transcription. And we first saw editing in HEC293 T cells at this HEC site 3. We found that the N-terminal fusion versus the C-terminal fusion of the MLV reverse transcriptase was slightly less efficient. So we settled on this C-terminal fusion of the RT to Cas9 H840A nickase and called it PE1. And as you can see in this graph, slightly extending the length of that primer binding site led to pretty dramatic differences in the efficiency of an edit that we were installing at a position right downstream of the nick site, a T to A transversion edit. And we could get low efficiencies of prime editing in HEC293 T cells with this PE1 construct. And knowing that there are things within this prime editing guide RNA, like the primer binding site, that can be optimized to improve editing efficiency. So what we next did was looked at the reverse transcriptase domain of the prime editor. So luckily, the MLV reverse transcriptase has been extensively engineered for basic research applications. And many mutations are known in the literature, in both the patent literature and basic scientific literature. And so we looked at a few of these mutations and found that the installation of, in particular, 5 mutations was able to increase editing efficiency sometimes up to 40-fold between the PE1 variant and this engineered reverse transcriptase. So we call this second generation editor PE2, which again has 5 mutations in the MLV reverse transcriptase domain that very much improve editing efficiency. Another aspect of the prime editing system is the PEG RNA. So this is the guide RNA that contains the template for edits. And we found that the pegRNA could be optimized by changing the length of both the primer binding site, but also the reverse transcription template. And certain combinations of these lead to very over 10% editing efficiency, sometimes up to close to 30% editing efficiency, depending on the edit. And so, as you can also see in these graphs laid at the top, the PE2 variant of the editor performs much more efficiently than the PE1 version and is also more tolerant of different primer binding sites. So we've identified now 2 areas of the pegRNA that we can optimize to increase editing efficiency. The last thing that we did was consider the pathway of DNA repair for getting this edit to incorporate into the DNA, into the genome. So to quickly go through the pathway that we think of how this works, the first step is that the starting DNA sequence is going to be acted upon by the prime editor. You're going to generate a new 3' flap sequence that contains an edited strand. What you hope does not happen is that that 3' flap is excised and that the resulting nick is ligated and you go back to the starting DNA sequence unchanged. What you hope instead happens is that the new DNA sequence can replace the original DNA sequence, and then that 5' flap sequence can be excised, and the resulting nicked DNA can be ligated to produce this heteroduplex of DNA. And at this point, the cell, the mismatch repair machinery of the cell, has sort of 2 options. It can change the original strand to create the edit, or it can revert the edited strand back to the original strand. So what we do in a system that we call PE3 or PE3B, which is a variant of the PE3 system, we use a second guide RNA, this time a simple single guide RNA, to target and nick the unedited strand. And what this does is it ideally drives repair of the DNA towards the unedited strand so that we use the edited strand as a template for repair. And in this manner, we can install the edit on both sides of the DNA double helix. So this is the concept behind the PE3 system, and it very much resembles the concept used by base editors, which nick the unedited strand to drive editing efficiency up. So just to very quickly show, we tested this concept at several different sites in HEC293 T cells, and in many cases, we found that the introduction of a second single guide RNA that targets and nicks the non-edited strand somewhere downstream generally results in higher editing efficiencies. However, this also comes at the cost of increasing the levels of indels. So for example, on this, at this RNF2 locus, without any nicking guide RNA, we see only about 10% editing with the PE2 system. However, when we supply a guide RNA that nicks roughly 40 base pairs downstream, editing efficiency goes up roughly 4-fold to 40%. However, the indels also rise pretty significantly. And this varies very much by the site. So one of the things that we encourage people to do if they're trying to use prime editing is to sample many different sites for nicking, because how many indels are produced and the increasing in editing efficiency very much depends on where that nick is at that site. And so we were able to demonstrate that prime editing can make all types of changes to the genome. We were able to install every single type of point mutation, whether it be T to A or C or G to A or C or T at multiple positions in this 8 base pair stretch of DNA at this HEC3 site in HEC293 T cells. So we can very efficiently convert these bases. We can do this at many different loci. We can install deletions of up to 80 base pairs. We can install small insertions and deletions. So these are showing us 1 or 3 base pairs insertion or deletion. And we can also do combination edits. So we can make at the same time an insertion and a point mutation, or a deletion and an insertion. It's very versatile in the sense that whatever you program in your edit locus or in your editing template, in theory, could be transferred to the DNA target site. And one other very exciting thing that I think prime editing does is it allows you to potentially install edits very far from the nick site. So we're all very familiar that SPCAS9, for example, is sometimes limited in its applicability because of the NGG PAM requirement. And there may not be an NGG close by to where your edit that you'd like to make is located. So prime editing in principle can be used to edit target nucleotides very far from the nick site. So We've tested this out to as far as 30 or more bases from the NIC site, and we still see quite efficient editing at this particular locus, up to 30 to 40%, even 30 bases away from that NIC site. So this opens the door to many possibilities where sort of by comparison, HDR is quite limited by the efficiency of installation of the edit once the NIC site or the cut site is more than roughly 8 to 10 base pairs away from the edit site. We were able to demonstrate that prime editing can work in various cell types, human cell types, so K562, U2S, and HeLa cells, albeit this occurs with lower efficiency than in HEC293 T cells. And also very exciting for me is that we were able to show in mouse primary cortical neurons, which are non-dividing postmitotic cells, that we also see prime editing. So sort of implying that replication is not required for efficient prime editing, sort of contrast with homology-directed repair, which in general does require cycling cells. And so what we'd really like to do is demonstrate how this could be used. So what we did was we used prime editing to both install and correct pathogenic mutations. So we did these experiments in HEC293 T cells. So we installed the A to T transversion mutation that's responsible for sickle cell disease. And then we corrected that same mutation using prime editing by just simply changing sort of the design of the guide RNA so that now we're making it invert that base pair back to the wild type. We also were able to do this for the 4-nucleotide insertion that causes a large number of Tay-Sachs disease cases. So we both inserted it and deleted it again for correction. And lastly, we installed a G to T transversion mutation in PRNP gene that confers resistance to prion disease. Hopefully through the talk, I've sort of been able to describe to you how prime editing is this new genome editing strategy that works by directly copying edited DNA into the genome, and it doesn't require any double-strand breaks or donor DNA. The template for editing is carried on the RNA instead. PE1, PE2, and PE3 were 3 different systems that we developed, each successively offering higher editing efficiency in human cells. And we were reaching upwards to 20 to 50% on average of editing in HEC cells, and we observed very few bystander edits in the sense that nucleotides that are adjacent to our desired edit are not necessarily modified. We have low levels of indels on the whole, and we see very little off-target editing by comparison to Cas9 nucleus. I didn't show that data, but this is another sort of feature of prime editing that I think is quite an advantage. And finally, prime editing scope is quite broad. So you can make edits very far from the PAM site. You can install different types of edits like transition mutations, transversion mutations, insertions, deletions, or combinations thereof. And in principle, you can use this to correct roughly 89% of this pie chart. Of course, there are challenges with actually carrying that out based on delivery and other things. However, in principle, prime editing can make the types of changes that account for all of these different classes of mutations. So with that, I'd just like to acknowledge the Liu Lab and Professor David Liu for all of the outstanding work that we do there, and the great people that I get to work alongside, and then this group of people shown on the left who made very significant contributions to all the work that I just showed you, and then also the funding sources. And with that, I'll take any questions.
Okay, so if you have any questions, please address them in the Q&A, and we are ready to hear from you. All right, so we have a couple of questions. Off-targeting is always an issue. What can you say about this issue, Andrew?
Yeah, so I very much agree with that, especially for therapeutic applications. We're very seriously, we need to think about how that would affect the implementation of these strategies. So I'd like to just quickly show maybe a slide that hopefully demonstrates why prime editing might be might have fewer off targets. So sort of the prime editing mechanism requires 3 different hybridization steps between nucleic acid strands. So one is the spacer and protospacer hybridization. This is common to all Cas9 based mechanisms. And then secondly, the primer binding site guide RNA, peg RNA needs to anneal to the nick DNA strand. So mismatches there could potentially, you could imagine, affect the efficiency of this annealing and initiation of reverse transcription. And then finally, this is somewhat gets you away from the spacer region. The sequence downstream of the edit, where we incorporate the homology to the locus, also has to match the endogenous DNA sequence. So even if the spacer mismatches, or there's an off target where the spacer is matched, the downstream sequence is not necessarily also going to be the same. So that's another level of discrimination in the mechanism. And so what we actually see is that by comparison to Cas9 nuclease, which I'm showing you up here, prime edits have very few off targets. So this is the top row here showing the on-target editing efficiency, and then the 4 rows below that are different off-target sites for these 4 different loci or guide RNA sequences, I should say. And we can see that by comparison to Cas9 nuclease, prime editing produces very few off-target edits, with the exception of this one off-target site 3 at this HEC4 locus, where frankly Cas9 nuclease is also much more efficiently cutting that sequence than the on-target site. So we think this is a really nice feature of the prime editing system.
Okay, so are you ready to move on to the next question?
Yeah, I can move on to the next one. Okay. So I can, comparing the efficiency of base editing to prime editing. Right now, base editing, I would say, is on the whole generally more efficient than prime editing. The tools have, first of all, been optimized for a few years. The comparison that we did in the paper, though, shows that in some cases, prime editing can be comparably efficient and can also produce cleaner edits. So one of the challenges with base editing is that if there are other target bases within the base editing window, those nucleotides can also be modified and you can get these bystander base edits. So with prime editing, I think one of the major reasons to think about prime editing when you're considering a transition-based mutation is the adjacent nucleotides in the surrounding region. And if you're in a particular region where it's enriched with potential bystanders, this is where prime editing could be potentially a better approach. So I hope that answers that question. The largest size insertion compatible with prime editing. So we've done insertions upwards to 40 to 50 nucleotides rather efficiently. And we don't know if there's an upper limit to this. One of the challenges we think associated with long insertions is that you're making the edit template longer and longer as you do this. And as a result, the sort of peg RNA is becoming more complex. The primer binding site is getting further and further away from the Cas9 complex. So the initiation of reverse transcription could be a challenge to overcome there, as could the repair efficiency of that new strand of DNA. But I would like to just remind you that the mechanism utilized by line elements reverse transcribes a 6 kb piece of DNA and incorporates into the genome. So in principle, I don't think there is a serious upper limit to this that's close to what we've demonstrated. However, there could be some technical challenges that still need to be overcome to do larger insertions. So somebody's asking about the mechanism of prime editing doesn't require double-strand breaks, but we do see indels, so that suggests double-strand breaks do occur. So we have done some work to reduce the indels that are generated by the PE3 system. That's called the PE3B system. I didn't show it, but what this does is allows you to make the nick of the that you're using with that single guide RNA be specific to the edit. So we're trying to intervene at this intermediate where there's a heteroduplex of DNA where one contains the edit, the Y is the edit, the other contains the original sequence, the X. If we make the spacer of this nicking guide RNA match to the edited sequence, but not the original sequence, in theory, you can temporarily restrict when that nicking happens. Because from our standpoint, we believe that the reason double-strand breaks occur with the PE3 system is that you're nicking at the same time both strands of DNA, which could lead to a double-strand break. So if you actually restrict that nicking till after the first strand has gone in, you actually see far lower indels. So the PE3B system, you can, for comparison, look at this +4 A to G edit at RNF2, where the indels start out about 8% and go to virtually nothing, and actually the editing efficiency increases. And then I think more dramatically at this +7 A to C edit at FANCF, where the indels are matching the efficiency of the actual desired edit around 20%. You use a PE3B system, again, the indels drop to a very low level. This is one potential strategy you could use. It does require another PAM sequence sort of on the on the other strand in the correct place, so nested PAMs. But when it's an option, I think it's a very good one. Okay, let's see. And I can't make too many comments about the efficiency in plant applications. I think there are definitely going to be different requirements in plants, and their DNA repair is likely a bit different than human cells. It was very encouraging though to see that these early attempts in plants have been successful, and I'm hoping further optimization will really make that process more efficient. So we have seen that edits can be made fairly far from the PAM. So we, as I showed, about 30 base pairs away. We don't know how far you can go. We don't know the upper limit yet. It might be related to the sort of the upper limits of insertions that we can carry out because, again, with a longer edit, you require a longer template. And so there just might be challenges associated with making PEG RNAs with very long templates. Maybe I'll take one more question. Can we create homozygous mutations using prime editing? You indeed can. So we did that for our paper where we made homozygous mutations in HEC293 T cells that carry the sickle cell mutation or Tay-Sachs allele. The HEC cells are on average triploid, so you have a little bit of a harder time making a homozygous edit in those cell types, but fortunately the editing efficiency was high enough. And you can sort for cells that express the editor construct efficiently. So it's definitely possible to do that. And other cell types where editing is lower, it could be more challenging to make homozygous mutations, but should be possible in principle. So maybe I can take the rest of the questions in the breakout room, and I'll hand it over, I guess, back to Mara.
Great, thank you. Thank you very much. I'd really like to thank the organizers for giving me an opportunity to speak to you guys today about a benchtop platform that we are developing for generating highly multiplexed variant libraries. Now, before I get started, I'd really want to make sure I give my acknowledgements out there. So I first of all want to thank the team at Inscripta, who I have the privilege of representing the great work that they have done here today. And I'd also like to make sure I acknowledge our collaborators. And I'll present some work today that we have been doing with John Stokes and Jim Collins at MIT, as well as some work we've been doing with Jeff Boca at NYU. Now, for many of you who don't know who I am, my background has really largely been in reading genomes, so genome assembly, variant identification. And so I've really had the privilege of understanding how reading genomes has really powered discovery in a wide variety of areas. However, one of the other things that has become really incredibly apparent is that you really just cannot get to the level of detail of genotype-phenotype correlation that you would like by only sampling natural variation. And so this is a figure from a recent paper on a database called GNOMAD that has been trying to systematically survey genomes and exomes. And so we can see they have quite, over 125,000 exomes and over 15,000 genomes, and this number continues to arise. And what they're seeing here is the percent of all possible variants, what they can actually observe in these populations. Now, we know that because this is really biased to exomes, so one of the reasons they can't observe some of this variation is because of selection. And they're actually trying to really take advantage of that so they can use this information to help interpret clinical variants. But we also know that one of the challenges is really just in the sampling bias that you see. And in fact, it is interesting, GNOMAD is trying to take advantage of the power of evolution to see if they can use that to help interpret clinical variations. And we have, historically tried to utilize this power to understand genotype-phenotype correlations. But the problem with most of these approaches, in fact, is that these are very slow, and they have, despite their power, some significant limitations, right? So even when you're doing, artificial selection or directed evolution, once you find that phenotype of interest, you still have to go into that genome and find that variation that's driving that phenotype. You can't really control the variant types that are being created, nor can you control the location of that variation. And the cycles of learning tend to be extremely long. Now, we know the challenge of this, and we'd all like to really do programmable variation. We're at a genome writing conference, right? So this is what we're all interested in. But many of our approaches for doing this to date are still somewhat limited. So we can think about looking at, going through large numbers of locations in the genome, but we're typically limited to the types of variations that we can do in those cases. Or we can maybe do a large number of edits, but often only at a limited number of locations throughout the genome. And what we'd really like to do to power discovery is to get up into this quadrant where we can make a large number of edit types all throughout the genome. And this is really something that we think is going to power both genome engineering as well as genome discovery. And so this is why we have really focused on developing a platform that allows researchers to generate large libraries of programmable, trackable variation. And so with that, we'll introduce the Onyx platform, which is really a platform that combines software for doing your design, as well as instrumentation with some novel microfluidic devices to help manage cell handling and normalization to produce these very large-scale engineered cell libraries that will allow you to make up to 10,000 edits with a single edit per cell with a very diverse type of edits. And so the specifications for the platform. So currently, this platform supports either E. coli or S. cerevisiae. Now we will provide some standard strains that we have characterized, but we'll also support customer strains. And we allow you to do insertions, deletions, or substitutions, where you can now make libraries of up to 10,000 edits in E. coli or up to 6,000 edits in yeast, where on average what we're seeing is libraries where about 30% of the cells are edited, but we can see a wide range depending upon the type of library. Because what you're doing is you're going in and designing the experiment you want to do, it will take us 3 to 4 weeks to make your custom reagents. But then once you get those, it's only about 1 to 3 days of instrument time, and then however long it takes you to do your phenotyping regime. And so what we think we've allowed now is the ability for us to really start thinking about programmed evolution. So we can take advantage of this powerful approach, but with fewer limitations, right? So because this is programmed variation, you know where that variation is in the genome. And because we barcode our reagents, we can help direct you to that location more quickly. We can control the variant types as well as the location. So this means we can increase the cycles of learning and really start generating large amounts of data. So one of the ways that we have been able to do this is by innovating on chemistry. And this also really allows to drive home the point that what we're talking about here are really precise edits. And so historically, this has required some sort of partitioning approach where the guide RNA and the homology arm have to be added to a partition. Often this happens in 96-well plates. But because of innovations made by scientists such as Andrew Garst, who is one of our founding scientists, we've now covalently linked the guide RNA with homology arm. And we've also added this barcode that allows us to track these reagents. So now we can really think about using the cell as the partition as opposed to using a well or any other physical device as a partition. And so part of this means we also need to really start thinking differently when we think about things like edit rates or library composition. And this is because when you have just a single edit, it means your edit rate and your library composition are really the same. So if you have a design that edits at about 50% efficiency, about 50% of the cells in your library will have that edit. And so that corresponds very nicely to your edit rate. But when you now have a library of designs, it's not likely that you're going to get equivalent edit efficiency at all loci. We've just heard even in some of the previous talks how you get different editing efficiency at different loci. And so you really need to start thinking about different metrics in terms of thinking about characterizing the output, right? It's about the library composition. And so there's really sort of two things that you want to think about. So in this cartoon, we have our output tube of cells where the round guys represent unedited cells and the square guys represent edited cells. So one thing that, is pretty obvious, you care about the fraction of cells in that population that are edited, right? And so in this case, you're really just counting the shapes. But what you also want to care about is the diversity of those designs that you see in that library, right? Because you can have 50% of your cells edited, but if only one of your 10,000 designs is represented, you still probably don't have the library that you really want to look at. So you also want to take a stab at understanding how many of those designs that went into your library are represented, and this is what we see here in the colors. This is also because of now thinking about these library dynamics. This is why that we've really had to innovate on the micro, make sure these library dynamics are controlled and we get nice, reproducible, even libraries coming out the end of this process. So now, because of this power and scale of being able to produce these types of cell libraries, we really think this turns on a wide variety of applications. And this is just really a small subset of those. And what I'm going to do for the rest of the talk is tell you about two of these applications today. Now, the workflow for both of these applications is relatively the same. So we can use the software to design our edits, and then we generate our library of engineered cells. We then apply some sort of growth selection. In this case, we're going to sequence using what we call our barcode diversity assay. What this means is we're going in and sampling the barcode that we see on those reagents, so we can get a sense of that distribution throughout the library over the course of the experiment. And then I'll show you some of the data that we've seen coming out of this. So the first story I'll talk about is work we're doing with John Stokes and Jim Collins at MIT, where they're really interested in exploring new avenues for antibiotics and new ways that we can think about applying antibiotic treatments to control infectious disease. So, you know, in general, when we think about antibiotics, we tend to think about, you know, the specific thing that that antibiotic is doing, right? You know, and they fall into these classes of antibiotics that maybe inhibit DNA replication or transcription or translation. But in fact, you know, even though we tend to think about their function in this very narrow way, they're really often having very large systemic effects on the cell. And so what we'd really like to do is take a much more systems level biology approach to understanding the full level of impact of these antibiotics. But I would say it's not even just about understanding these mechanisms of action, because we also know in this arms race with antibiotics that there is there's always the cell coming up with ways to counteract the antibiotic. So we also want to try and think about understanding the modes of resistance. And so by really taking the systems level approach, we'd like to start getting into aspects of both of those things. And so for this experiment, we actually made two libraries, and I'll give you a little bit of the details of both of these libraries in the next slide. We applied two different antibiotic treatments with two different regimens in this experiment, and then we assayed these by our barcode diversity assay. So for these two libraries, these two libraries are actually pretty modest in scale for what we can do, but this was a pilot that we really wanted to try. So in the first library, we targeted a little over 590 genes, with 230 of those having been annotated as essential in EcoPsych, and we'll talk a little bit more about that. The second library, we really tried to focus on genes for central metabolism. Now, for both of these libraries, we made gene knockouts, and to do these, we introduced triple stop codons to stop transcription. And then we also did regulatory variants. So we were able to take advantage of the iGEM standard parts library and identify these promoters that have been assigned different relative strengths based on their performance in a plasmid system driving a reporter gene. So we can take each of these promoters now and insert them in front of all of these genes throughout the genome to understand how these different promoters might impact the cellular phenotype. And so one of the things that was, we were really excited, and I think the Collins group was really excited about, was now this new ability to really interrogate essential genes. So unsurprisingly, when you look at the knockout variants, we really see, and we're counting the number of times we see these reagents, we really see a depletion of these edits in the essential genes. And this is not surprising, because if a gene is essential and then you knock it out, you would expect the cell to die. But when we look at these regulatory or promoter variants, we can see a much better recovery of these essential genes. And so this gives us now a mechanism to understand how at least regulating their expression can have an impact on cellular phenotype. So what this data is showing is both the nice correlation between technical replicates. So we see when we compare controls, we see a strong correlation. And we actually don't see much change when we look at low concentrations. In this case, we're looking at gentamicin. And then when we look at the high gentamicin, we see, when we compare the controls or the high concentration, we see good correlation. And then we start to see skewing in these other, comparing the high concentration of gentamicin to controls. And this is what we would expect, because this antibiotic treatment is one of the things that's driving the phenotype. And so this is just one way to think about how we can use these sort of counting assays as a proxy for phenotype. So what we have here on the x-axis are the concentrations of the various gentamicin treatments. And on the y-axis, we have the count of barcodes we see for that sample. And in this case, we're only looking at one gene, but you can do this for every gene in the genome. And what we really do is we actually think about using this change in slope as the biological response. And one of the reasons that we want to do this is that it allows us to sort of normalize for the different starting counts that we might see in that library due to just the different random stochastic distribution of designs throughout the starting library. I think one of the other exciting things about this, so as a human geneticist coming in to think about microbial biology, I think I was still surprised to understand how many genes in the E. coli genome still had not been ascribed function. And so this so-called Y-ome, or these Y genes, we're starting to pull out information on those and be able to assign those to pathways and phenotypes as we see a response in this gene with treatment of gentamicin, which when knocked out, we see an increased sensitivity to gentamicin, and as we increase expression, we see increased tolerance. We're also able to sort of dig into the data and sort of look to see if we can recapitulate known biology, right? So gentamicin is pretty well studied, and we see that we can recapitulate. We see when we knock out this ACE-B gene, we see increased tolerance to gentamicin, and this had previously been reported. And lastly, we can really think now about looking genome-wide. So in this plot, we're looking at all of the genes in the genome, and the blue dots are places where genes have a significant change. And interestingly, even though Cipro and gentamicin have really different modes of action, where Cipro inhibits DNA replication, whereas gentamicin inhibits protein synthesis, we can find some genes that have common impacts on both of these pathways, and then other genes that actually have impacts that are specific to each pathway. And so we can really start thinking about how we can both look at mechanisms that are common, maybe more due to cellular metabolism, versus genes that might be impacting each specific pathway. So next, I'd like to talk more about some work we've done on strain optimization with Jeff Bulka on his yeast synthetic chromosomes. So I'm sure, as most people at this conference are aware, Jeff has been leading this SC 2.0 project where they would like to make completely synthetic yeast chromosomes. And I was fortunate enough to be at the GP Wright conference late last year where they announced essential completion of this. And so, one of the reasons they really want to do this is they want to make a complete synthetic controlled yeast genome, which will allow them to answer really interesting and important questions around genome biology. But an important aspect of that is that these synthetic genomes really need to grow at the same rate as wild type. And that's one thing they observed, that even in a yeast chromosome that only has one synthetic chromosome, they see some growth defects. And so Jeff's lab went through and did some manual work to try and start identifying variants. They had some candidates. They could identify 4 variants that when they changed these, reverted these back, they could start seeing some improvement in growth performance. And so what we wanted to do is see if we could take all of the candidate edits and put them all in one library and just assess everything at once to see if we could find the variants that were leading to the growth defects in these synthetic yeast. So again, in this case, we made one library. Again, because we had a relatively modest number of edits to make, we were actually able to make multiple redundant designs to look at these. The growth selection this day is just growth in rich media. And again, we do our barcode counting assay. So this slide just gives you a sense of the breadth of edit types that we were able to do. So in this one library, we're really able to look at all kinds of different edit types, SNP changes, insertions, deletions, to really get the scale and scope of this library. And luckily, we were able to, in one experiment, both recapitulate and find the variants that Jeff's lab had found previously, but also we were able to identify a score of other variants that are now candidate variants that Jeff's lab can go in to now verify that these are changes that impact growth in their synthetic strain. So hopefully today I've been able to convince you that the scale, performance, and access on this new platform Both allows us to rapidly recapitulate known biology, as we saw both in the increased tolerance to gentamicin with the knockout of the ACE-B gene, and confirming these variants that Jeff's lab had found that were impacting growth of their synthetic yeast strain. But we're also able to rapidly uncover novel biology. And I think really excitingly is that we're able to provide novel ways now to interrogate essential genes via regulatory changes. And so I'm really excited now to see how writing genomes will supercharge discovery in a wide variety of areas as well. With that, I'm happy to take any questions.